Good morning everybody. So as we are proceeding towards the end of this course, where we are now discussing in an elaborate fashion the electrochemistry and how it is applicable to analysis or development of new techniques in analytical chemistry. So analysis, and also today we will see, and we have also given you some examples for that how this particular part, that means the electrochemistry, can be beneficial to handle biological samples such as proteins, metal enzymes, etc., as well as the environment and the materials. So, in a sense that the whole gamut of analytical chemistry, what we are talking so far and will be talking in the next 3 classes, is based on something where we talk that we are doing some analysis and is the methodology development or some analysis of some real life samples that we will probably discuss in our last class, that how you tackle or handle the real samples. So, the analytical chemistry, what we can have and what we have discussed so far, this particular part is very important, because this, when we attach for, depending upon the sample, basically when we go for say analysis of blood sample, what we put, we make this as the clinical one only. When we go for these materials, we make it as the analytical chemistry aim to analyze the materials, particularly with the development of any such films and all these things which are required for your electrochemical measurements can come under this category. And as we are proceeding towards the end of this course, you must have the typical realization that in all cases chemistry is not playing the major role or the important role. It is a thing which can be developed, which is based on the basis, the fundamentals is on chemistry, but slowly you can apply to all branches of science, all areas of science, even history, geography, geology, all these things. So, slowly we are, as we finish the course, you will have the realization that whatever you have studied in this particular course, the basics and the fundamentals we have started from the school level chemistry knowledge, then we applied what are the analysis and what are the different analytical techniques we can have. And now as we move towards the end of this course, it can consider this as a typical analytical science. So, is a separate branch of science, definitely we can consider, where we can have the typical foundation we can have, because the most important area what people are talking about, the environment, when we deal to tackle the wastewater problem or the effluent water from the industry, the river water, the sea water and on the lake water. So, talking all these things, so environmentalist also can think of this thing, but without going for a very good or sound knowledge of chemistry, they can tackle these problems by simply saying that okay, whatever I am handling is based on your analytical science. Okay. So, what we will now go for is your biological part, that how we can apply this for your bioelectrochemistry studies. So, bioelectrochemistry studies will be definitely very important, as we have seen that some part we have exposed also during the development of the sensor, because these are very much interrelated. That development of the sensor, the corresponding fabrication of the electrode, and what are the solution we basically handle. So, the basics and the fundamentals, what makes your knowledge of analytical chemistry and electrochemistry in particular, and now the bioelectrochemistry, is that it should be very easy to understand if we consider something related to the reaction, what we are thinking or what we are talking about, the two electrodes, the cathode and anode. So, for the molecules which are coming from the living system, And the electron transfer reactions, basically, as I told you, that whatever you have, that what we want to burn, the food material what we consume. So, that is the result. This particular branch is also applicable to your food chemistry, agricultural chemistry also. So, food we are trying to burn to get carbon dioxide and water with required amount of energy elimination. Why we require that energy? Because we have to synthesize ATP molecules from ADP. So, this is basically known as the corresponding phosphorylation reaction, but it has some energy demand definitely. So, your phosphorylation reaction. So, what we have seen for our electrode reactions, that we can get these like this sort of thing, what we all know now. These sort of things. Okay. So, these are basically the thing what we can have then for your O2, because we will be burning your food material with that of your O2, that we know that how you synthesize your carbohydrates and proteins and fats also, and at one time you have to burn this. So, O2 will go for 4 electron 4 proton reduction to give you 2 molecules of water. So, this is the ultimate thing, and in between what you can have all these things are related to something what we call as electron transfer chain. So, we have the electron transfer chain. So, as we all know that this is also well known to us, where O2 can be reduced to water molecule, and we all know the corresponding E0 value is 1.23 versus NHE, and it is sometimes the corresponding O2 if we want to measure by cyclic voltammetric technique, that also give us some idea that with respect to your SCE or the silver chloride electrode, it will come close to 0.8 volt. So, this magnitude, so O2 is a very strong oxidizing agent compared to the material like food what we are trying to oxidize. So, during this electron transfer chain, we can have certain mediators what we have discussed in considering the corresponding, when we are considering rather that how we develop a particular electrode for the sensing mechanism of O2 or the sensing mechanism for your glucose. So, your electron transfer chain is there and it slowly goes like this. So, this is the reductant basically which can be oxidized by these, and at different steps of these in this electron transfer chain, you have the corresponding amount of the corresponding E0 values for this couple, because all these are attached with what? Your oxidant as well as the reductant. So, is the pairwise. So, pairwise it can be your mediator, it can be your enzyme. What we have seen that their glucose oxidized in the oxidized form and the reduced form when it accepts the electron. So, these things are there. So, these all are coupled with these E0 values. So, you will have for all these E0s. So, successively as we move from left to right, E0 values are increasing. So, when we come to the left, basically your E0 value is very less, that means your food material attached to this particular oxidizing agent is not so exothermic in nature. As a result, we can say that your food is not getting burned. So, in a regular fashion, in a very systematic manner, we try to oxidize the food material in a stepwise manner, such that all these things, particularly these are basically the different cytochromes, the cytochrome B, the cytochrome C and cytochrome C oxidase also, CCO we call. So, at the end we have the cytochrome C oxidase, so which is a very complex molecule for all these things. But in terms of these E0 values, because these can be labeled depending upon, one is the corresponding classification for these molecules as their structures. So, B, C and CO, that means cytochrome C oxidase in terms of the different structures, as well as you can have the different E0 values. So, we get the progressively your E0 values will increase until it is reaching the corresponding dioxygen water couple. It reaches the dioxygen water couple. So, we can consider these as the good redox mediators. So, these good redox mediators which are coupled nicely such that required number of ATPs are also synthesized during this transfer. So, what is this particular example? What is told in terms of the corresponding language, that electron transfer between molecules. So, the molecules are now your mediators like cytochrome, the different cytochromes in the cytochrome electron transfer chain at interfaces. So, when these are basically attached with these, so this is there. So, you have the corresponding one which is accepting that electron from that particular food material. So, this is getting oxidized form and this is getting the reduced form. So, reduced form is again attaching with that particular species which is getting oxidized. So, oxidation reduction keep on going from left to right. So, is the interface, the central role is playing for all living systems, because you require the electron transfer, you require the energy development or the energy currency by burning of the food material such that we can have the required number of ATP molecules for that particular process. So, when we talk in terms of this electron transfer, so definitely we can talk in terms of the bioelectrochemistry, which can be a study and application of biological electron transfer processes. So, it is a biological, typically a biological electron transfer processes what we can have, and is definitely is study how the electron transfer is taking place and how we can apply also. Once you know that we can monitor this electron transfer in terms of your food material or oxygen reduction, we can have some good applications also, and two very small applications we have seen for the development of the sensors. So, all these different electron transfer processes initially we can monitor for their thermodynamic electron transfer potential values, that means the E hub values or E0 values for their cell, what is formed between the cathode and the anode for that particular electron transfer reaction. So, when these are there, so when they are playing some essential role in all living systems. So, when this that means that is playing a central role in biological systems, it can be essential for the processes in which biological cells captures and use energy. So, how the energy is getting and how you use that energy, typically from the burning of the food material or any other material which is typically a reductant or a reducing agent. So, your food material is typically a reducing agent, your carbohydrate, your glucose is the typical reducing agent which can be used for reducing some other groups, and then ultimately will be going for the reduction of your dioxygen molecule, which is the ultimate oxidant. So, these things are basically can be keyword, your electron transfer, your bioelectrochemistry, and one thing is also their electrocatalytic thing, because these cytochromes and all these are we all know that some of them are containing metal ions. So, they are metalloenzymes we call. So, this cytochrome C oxidase is also you have iron center and you have copper center. So, iron 2 plus is settling between 2 plus and 3 plus, and this is between 2 plus and 1 plus. So, this basically goes for these things. So, what we can label these as typical metalloenzymes. So, these metalloenzymes, so we will take out this part, that means they are enzymes, definitely by definition they will be enzymes, and we can talk their nature in terms of the enzymes. And these metal ions are responsible for electron transfer, electron transfer. And if this is a typical cyclic process, that means is going to oxidized form and coming back to the reduced form, as we find by determining the cyclic voltammetric pattern or the potential for this electron transfer reaction, and it can go for n number of cycles in this particular process. So, you have enzymes, you have electron transfer. So, it can be termed as electrocatalyst also. So, our idea behind all these studies and knowing the corresponding applications is that how we can get a catalyst which can show some reversible electron transfer reaction. So, we have to develop some catalyst, is not some other catalyst we know that is not going through electron transfer reaction. So, they are redox catalyst or the electrocatalyst, because the development of all these things can solve so many problems related to electron transfer, and we have to develop this good material as the catalyst. So, we get the idea from the biology. So, biological electrocatalyst we see little bit, and if we can have some hybrid mechanism that you can get the material or the biological electrocatalyst from the biological or the living world, you can apply that to some synthetic mechanical thing, that means some electrode or the some cell which we can develop in the laboratory. So, one such example, then again we are talking about the oxygen to water conversion, that means the 4 electron 4 proton reduction of O2 to 2H2O. We can see that for that electrocatalysis for the 4 electron reduction of oxygen to water at neutral pH remains a key barrier to the development of efficient polymer electrolyte membrane fuel cells. So, now we are talking in a single line so many important lines to be understood, that if you have the idea that we are getting all these informations from the biological world, the electrocatalysis for very simple substrate like you are sensing that O2 molecule as your Clark oxygen sensor, but at a neutral pH is not that it is driven by acidic pH or the basic pH, it remains the key barrier for the development of efficient PEM fuel cells. So, we want to develop a fuel cell such that which can use that living systems, that means the biological molecules as the corresponding fuel for the cells, and a polymer electrolyte membrane, because membrane we already know now how we can use the membrane on the electrode surface, which can allow preferentially some to go and some should not. So, the development of these PEM based fuel cells can be an immediate target for those things where the pH is a neutral pH. So, it's definitely we have the target, we have the problem in our hand, and how we can develop all these things, because definitely you have to try for a system for n number of times, then only you achieve something where we can go for a very useful membrane type of fuel cell which can be used for generation of your electricity, the future electricity. So, biological electron transfer, that is why we are pushing it to a biological system. A biological electron transfer reactions are very important, and already we have seen in the development of biosensors. Now, we will talk little bit about the biofuel cells, and if possible, if time permits, definitely you can have, but at least you should know at this point that you can have some synthesis, as we know in the chemistry terminology, that when we make a molecule we go for A plus B, the target is C, and sometimes some other part is also like water molecule, like carbon dioxide is coming out as the D or E molecules. So, synthesis is there, but if it is going for electron transfer only, then electron transfer can take place, and then bioelectrosynthesis is there. Biological molecules can be modified by electron transfer reaction to get some useful synthetic molecule which can have application as medicine or some other good purposes. So, this biological electron transfer can be tackled by something where we see that one particular terminology you should be able to understand, that nicotinamide adenine dinucleotide, that reduced form NADH we call. So, these are basically the cofactors in the biological world. So, this dinucleotide in the reduced form can show some reactions where it is dependent. So, oxidoreductase enzymes. So, you have the oxidation reduction reactions. So, both are coupled together. Instead of saying them as the redox enzymes, we call them oxidoreductase enzymes. So, these redox enzymes are NADH dependent dehydrogenases, or redox proteins can be exploited in the development, not in the development of all these things. So, like your glucose oxidase, your NADH is some other molecule, and if it is in the reduced form, definitely you should know that this is the reducing agent in the giving you the corresponding mediator business. And one important mechanism or one important characteristics will be useful there when we talk in terms of the electron transfer reaction. Now, at the same time, if electron moves in one direction, in the opposite direction you get the proton movement. That is why the corresponding reduction of this O2 molecule, what we are talking here is that if we say that it is a 4 electron transfer reaction only, is not that you should also have the 4 proton transfer. As a result, you get 2 water molecule from your O2. It is not that you only go for your O2 to 2 O2 minus. You have to take these 4 proton to get these 2 water molecules. So these two are coupled together, and if we find that in one end you have the transfer of electron in one direction, you have the transfer of proton in the other direction. So, electron in this direction and proton in this particular direction, and most of the cases you have this a coupled one, because if you have the very simple functionality, as we all know, that CONH function is the backbone of all these things what we are talking here in terms of the corresponding protein, polypeptides and all these. So, these are all your peptide bonds. So, if you have the peptide bond and if you are able to go for the electron transfer reaction on the peptide bond itself, or it is bound to some other species like metal ion, what we see there that during that particular reaction you can go for, that means the O is there. So, that means you have this O, and if it is in the oxidized form, so that means you take out the electron, you have to take out the proton also, because this can also go for the loss of proton also. And sometime it can also show that the amide protons in the charged form, and that the charged form can go for your electron transfer reaction. So, whenever there is available proton, it can go for proton loss or it can go for proton acceptance during the electron transfer reaction. So, like electromotive force, we all know the E0 values and E-half values, we all know that the electron transfer potential or the electron transfer sensitivity for a particular system and how the driving force is there. So, it is a force basically. So, proton can also have some kind of that kind of force. So, we define it as the proton motive force, and now we can have something where we bring, because earlier we get the exposure for your membrane which is very close to your electrode also. So, where we get this proton motive force? The proton motive force is a very complex one and a combined form of a potential difference. You should have a potential difference from one side to the other, like that of your metallic rod dipped into the solution, we have a potential difference. So, the across the membrane and the difference in proton concentration across the membrane. So, all biological membranes we all know that your ATPases are there, then the potassium ion, sodium ion, all these are enclosed in some membrane. So, lipid layers are there and the membranes are there, and if the proton concentration on one side of the membrane to the other is different. So, if there is a difference in proton concentration for this is across the membrane, what we get? Definitely we will get a proton motive force. So, that is the corresponding idea behind defining this proton motive force. So, definitely you have a different in proton concentration like that. When you talk about the electron transfer reactions and the electromotive force or the EMF, basically the electromotive force we talk, that means we know that some way either from the downhill or from downhill to the uphill electron can be moved. So, the electron can be transferred. So, when the membrane is available and you have two different proton concentration from left to right, so proton will allow that particular proton to traverse or move from one part to the other such that we reach to a equilibrium, because on all these cases whatever we are talking about is a non-equilibrium condition. So, that non-equilibrium condition we have to establish in terms of either a EMF value or a PMF value. So, both these things, that means both contribute, that means the potential difference across the membrane, you must have a potential difference across the membrane at electrodes when it is there. Those electrodes can also contribute to that particular potential difference, and your difference in proton concentration both can contribute to the available free energy change or available free energy for a particular type of reaction to go. So, these bioelectrochemical systems thus we have in our hand, and we should know now how they work. So, these bioelectrochemical systems, if they go for electron transfer reactions, they can give rise to, because already we told you that you can have the biofuel cells. So, what are those biofuel cells? So, when you go for little bit of elaboration of those things, that means we can have microbes are available. So, these microbes are available for generating electricity. We get the fuel cells. So, you know, like battery, you have the fuel cell, we can generate the corresponding electricity for you. So, the microbes are utilized to generate this fuel as the corresponding electricity. So, this will be microbial fuel cells. Then another one is the opposite one, where we can go for electrolysis. So, when you go for the electrolysis, because these two are combined together, if we go for the fuel cell, somewhere you can have the electrolysis also, otherwise you cannot get this thing, like that of your electrolysis of water molecule. The water molecule, when it is electrolyzed, we know that we can get oxygen and hydrogen over there. But when we go for the fuel cell based on hydrogen, we know that hydrogen can be burned in presence of oxygen, because hydrogen is the fuel, because we do not have the natural supply of hydrogen, but oxygen is there in air. There are plenty supply of oxygen. So, this is hydrogen will be your fuel, and we can go for the corresponding hydrogen based fuel cell, but we should not very much bothered about the thing that where from we get the hydrogen. If there is a good source of these hydrogen, we can have the hydrogen based fuel cell, and if microbes are utilized, so those microbes will be there to generate the required amount of hydrogens for your cell. So, these are known bioelectrochemical cells. So, once you are asked that what is your bioelectrochemical cell, you can have the idea that okay, it is electron transfer reactions and the electrolysis cell or a fuel cell. And outcome of the two combined thing, that means one is the biological part and another is the electrochemical part. So, two processes can go side by side to generate electricity, to generate hydrogen or other value added chemicals which we need for other purposes. So, value added chemicals or some useful chemicals, as we get chemicals from petroleum, we call them as the petrochemicals. So, these microbials or the microbes can be useful for giving you the required amount of all these chemicals, including your hydrogens. So, two such cells we can abbreviate that MFCs and MECs. Two are common, the cell and microbes, only one is fuel, F is fuel and E is the electrolysis. So, the current what we can generate from a microbial fuel cell, so with the hope that we can generate electricity from there, is directly proportional to the energy content of the wastewater. Not only microbes, now we are correlating to something which we consider as the wastewater. So, it is a concern for the environmentalist, is a concern for the water treatment, how you get pure water, at the same time how you get the energy. So, this wastewater used as the fuel. So, there is no need to think of that where from we get that particular fuel. So, if you have the wastewater, you know that the organic matters are plenty in that wastewater. Only thing that you have to choose the right microbes which can degrade those organic matter, and these fuel cells can measure indirectly the corresponding solute concentration of the wastewater. So, in terms of its biosensing capability, we can use this to determine the wastewater concentration of the solute in terms of your O2. You can detect the glucose, you can detect some other species also. So, the biosensing part is over. Now, you know that some good amount of material is there which can be used as a fuel for generating electricity. So, those things can come to give you the corresponding development of microbial based fuel cells. Okay. Thank you very much, and we will continue this for our course for your next class. Okay. Thank you.